Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, for those who don't know us, so we will introduce ourselves. My name is Riris Ayu Trilianti, and my partner.
Hi, my name is Linda Mutia, and we are from Biology Education 2019. So, in this opportunity, we would like to present to you about permanent tissue.
Let me begin with some information on tissue. Tissue are groups of cells that work together to do a job in the body. They are a group of cells having the same origin, structure, and function. Tissue is different from organ. In a plane, the tissue is divided into two groups, meristematic and non-meristematic tissue. We can see from the chart that the permanent tissue is divided into two, namely the simple tissue and the complex tissue. Simple tissue is divided into three, namely parenchyma, collenchyma, and sclerenchyma, while the complex tissue is divided into two, namely xylem and phloem.
And now, I'll present to you about parenchyma. What is parenchyma? Parenchyma tissue is basic tissue found throughout all plant organs. And then, parenchyma has several characteristics. And first, originating from protoderm and ground meristem. Protoderm is on young leaf, and the ground meristem is on the root. And second, cells with thin wall. Third, most are living cells. Fourth, can be totipotent. Totipotent is the ability of a cell to be able to multiply itself in all possible developments. 5. Vary in shape. Isodiametric, similar in diameter, the cell may be oval, circular, or polygonal. As you can see in the picture, that is the shape of parenchyma cells. That A. Diagram of the orthic tetrahedron. A. 14-sided polyhedron. B. Diagram of a cell from the pith of Atlantis. It has 1 heptagonal, 4 hexagonal, 5 pentagonal, and 4 quadrilateral faces. A total of 14 faces. An example of a cell approximating an orthic tetrahedron. C to F. Diagram of pith cells of Eupatorium. The numbers of facts are 10, C, 9D, 16E, and 20F.
And next cells may be more or less elongate or variously lobed or branched. This picture shows several forms of parenchyma. First, there are polygons, secondly lobed parenchyma, and branched parenchyma. In the branched parenchyma, figure A is the star parenchyma in the Apheus Janus. Figure B is the parenchyma in the rhizome of the Acarus calamus. The picture shows the shape and wall structure of parenchyma. Parenchyma from the stem pitch of Betula. Figure A is in younger stem. Figure B is the cells have only primary walls. Secondary walls also occur. Figure C and D, parenchyma of the Erythema type. And next, the characteristic of parenchyma. Cells may be arranged in compact cells or with large intercellular space. The picture shows the parenchyma with clear branched cells, intercellular space between the leaves of Canna. Parenchyma be found in pith, mesophyll of leaf, cortex of stems and roots, flowers, fruit, seeds, xylem, and phloem. This picture shows the location of the parenchyma of the stem of the dicot. Parenchyma can also be found in sunflower leaves, which include dicot plants. Look at the picture circled in red, which shows the palissaded parenchyma and spongy parenchyma tissue.
And then be found in example given. Pith, mesophyll of leaves, cortex of stems and roots, flowers, fruits, seeds, xylem, and phloem. As you can see the pictures, here is the root image. Parenchyma is located in the cortex. Be found in example given: pith, mesophyll of leaves, cortex of stems and roots, flowers, fruits, seeds, xylem, and phloem. And next, the figure, that is cross-section of the petal near the spot. That is epidermal cells weakly colored, subepidermal cells strongly colored, mesophyll no color, and lacunae. And next, be found in example given: cortex of stems and roots, parenchyma with large intercellular space, and branch is known as irenchyma. The right picture, that is irenchyma of hydrophyte stem, and the below picture, that is irenchyma of hydrophyte root. And next, parenchyma cells may contain chloroplast, known as chlorenchyma. Colenchyma may also contain chloroplast. Both differ in some aspects. The characteristic of chlorenchyma, that is chloroplast-containing parenchyma cells. In isodiametric cells, chlorenchyma cells are not interlocked, soft uniformly thin cell wall, and last, photosynthetic and storage in function. And then the characteristic of colenchyma. Chloroplast may be present or absent within the cells. In transverse sections, the cells look elongated and angular. By their end walls, the cells are interlocked. Hard, unevenly thickened cell wall, and then photosynthetic and mechanical in function. And then the following are some of the functions of parenchyma tissue. There is first, storage of food, root and stem cortex, pith rays, xylem and phloem parenchyma. And second, to support aquatic plants to float on water, there is irenchyma. And third, photosynthesis, there is chlorenchyma, palisade and sponge in leaf. And last, parenchyma can be active to divide, example given, cork cambium, and help the plant in secondary growth, formation of cork, and healing of wounds.
The second permanent tissue is colenchyma. The meaning of colenchyma according to Greg is cola, which means thick glistening cell wall. Origin of colenchyma developed from soil meristems or from procambium. Example, graveolens. Colenchyma, a single cell type of simple tissue. Colenchyma has a shape ranged from round, elliptical to elongated cell. Colenchyma is more or less complexly arranged as the thickening materials deposit more heavily at the corners and on the radial walls.
Next, share similar characteristics to parenchyma, but thickened cell wall. The thickening is unevenly distributed. Gel wall composition, there is cellulose, hemicellulose, and pectin, but absent of lignin or in lignified cell wall. And last, outcapit the peripheral layers of cortex indicates and beneath the epidermis or below a few peripheral layers of parenchyma.
And then I will go on patterns of distribution of cell wall thickening. Number 1. Angular colenchyma. The thickening materials deposit at the angles or corners of the cells. In a cross-sectional view, the thickenings occur at those places where several cells meet, found in the petiole of Cucurbita and stem of S. tuberosum. The picture was a cross-sectional view of colenchyma in Salvia.
And next, lamellar or plate colenchyma. The thickenings at tangential cell wall, which are found in cortex of stem of Sambucus nigra and petiole of Cochlearia armoracea.
And next, lacunar colenchyma. The thickening materials deposit at cell walls which are in direct contact with intercellular space. Example, petioles of Salvia, Malva, and Asteraceae. In the picture shows these thickenings do not fill completely the intercellular space, and empty intercellular space can be observed.
And then annular collenchyma, which is defined by Duquesne in 1955 as it possesses circular cell lumen in transversal section. Annular collenchyma is collenchyma tissue whose cell walls evenly thicken. This type of collenchyma is rarely found because it is only found in carrot leaves and some vines.
This picture shows a cross-section showing the distribution of collenchyma and vascular tissue in several parts of the plant, namely the humerus petiolus, sambucus stem, mentastem, bastiana stem, and cucurbita petiolus.
And then what is the function of collenchyma? First, serves as mechanical support to the organs. Second, in some cases, the peripheral thick-walled collenchyma becomes thin and regains the meristematic activity. Example given, Pelogen, the curcumbium, which divides to form the periderm. Third, chloroplast existence involved in photosynthesis. And last, manufacturing sugar and storing it as starch. And next, we will present to you about sclerenchyma. After that, specific features there is usually dead cells or mature sclerenchyma, and then heavily thickened secondary walls and lignified or contain lignin, and then rigid cells or unstretchable, and next arrangement compactly or no intercellular space, and last, the main shapes, fibers and sclerates.
And then I will go on sclerenchyma fibers. Develop from meristematic cells, typically are long spindle-shaped cells with more or less thick secondary walls, and they usually occur in strands of lignification varies in type. Fiber is sclerenchyma tissue comprising of fiber cells. The fibers are present in hypodermis of monocot stems, in pericycle of many dicot, and secondary wood and hypodermis of many leaves. Fibers is economically precious that are divided into hard and soft fibers.
And then as you can see the pictures, that is a septate fiber from a phloem of grapevine or physit stems. The septae are in contact with the pith secondary wall. And B, septate sclerate from the phloem of Paraschia or Cactaceae, in which the septae are covered with secondary wall material. And the next pictures, there is primary phloem fibers from the stem of basswood, Tilia americana, seen here in both A, transverse, and B, longitudinal views. The secondary walls of these long, thick-walled fibers contain relatively inconspicuous pits. Only a portion of the fibers can be seen in B. Fibers typically are long, spindle-shaped cells with more or less thick secondary walls, and they usually occur in strands. And then the next picture, there is A, tracheate, B, fiber tracheate, and C, libriform fiber in the secondary xylem or wood of a red oak tree. Wood fibers are commonly divided into two main groups, the libriform fibers and the fiber tracheates. And then the picture is, as you can see, that is primary phloem fibers. Transverse section of stem of Linum usitatissimum showing position of primary phloem fibers. The cell walls of the extract-leaf fibers are often very thick. In the phloem fibers of flax, the secondary wall may amount to 19% of the sectional area of the cell. And next, B. Stone cells or sclerates. First, they are lignified, extremely thick-walled. They are generally found in hard parts of the plants, example given endocarp of walnut and coconut. The shape can be isodiametric, irregular form, and ramified. The cell can be single in plant tissue or in group in central tissue of plants. And then, in most plants, sclerate is a dioblast that is a distinct cell possessing distinct size, structure, and wall thickness among the other cells. And last, in Magnoliophae, the cells is found in most of organs. And then the next picture, there is sclerates or stone cells from fresh tissue of pear or Pyrus communis fruit. The secondary walls contain conspicuous, simple pits with many branches, known as ramiform pits. The most commonly recognized categories of sclerates are first brachysclerates or stone cells, roughly isodiametric or somewhat elongated cells, widely distributed in cortex, phloem, and pits of stems, and in the flesh of fruits. In the right picture, there is branch sclerate from a leaf of the water lily or Nymphea odorata, as seen in polarites alike. Numerous small angular crystals are embedded in the wall of this sclerate. The secondary wall typically appears multilayered, reflecting its helicoidal construction. Crystals are embedded in the secondary walls of certain species. Many sclerates retain a living protoplasm at maturity.
Let's now move on to types of sclerates. First, brachyschlerate or stone cells, isodiametric and located in phloem, cortex bark, and mesocarp of Pyrus communis. Look at the picture is a stone cell from the flesh of the Pyrus fruit. And number 2, macrosclerate or stick cell, which is elongate and cylindrical. It's found in the inner layer of seeds of Leguminosae. In this picture, there are sclerates called macrosclerate with thickening of the walls of the epidermis of Phaseolus seed ulcers. Number 3, osteosclerate or bone cell that elongate with large ends like humerus bones. The sclerate is found in hypodermal layer of some species of Leguminosa, fruit and leaf of Cerophytes. This image is a pulse-shaped sclerate with branched ends. This is an image of the mast tissue in the Hakean mesophyll. Number 4, astrosclerate or star cells. Star-like structure found in stem of Cerophyte and petiole and mesophyll of Camellia sinensis. This image shows the astrosclerate of the Trochodendron stem cortex. And the last, trichosclerate. That's long branch sclerate with point ends buried in intercellular space. It's found in hydrophytes that are floating organ of Echinonia crassipes, root of Nymphaceae, and petioles of Pistia. This image shows the shape of the trichosclerate.